Alright. So this is the second problem. The DNA content of an individual cell undergoing meiosis can be measured by applying a DNA-specific dye to the cell and then passing it through an instrument that measures the staining intensity. The more intense the sample is, the more DNA it contains. A new cancer drug was tested on a population of rapidly dividing tumor cells, and their DNA contents were analyzed and compared with those of untreated cells. Here's the DNA intensities, and this is the percentage of cells in each corresponding to each DNA intensity by untreated and treated.
Okay.
And so the first thing I did was use the sentence that I underlined: the more intense the sample is, the more DNA it contains. Let's just start with the first question. So,
sure.
Plot percentage of cells versus dye intensity for the treated and untreated cells. Label the axes and indicate treated and untreated samples on the graph. So this is the graph.
Okay.
And then the percentage of cells would be the y-axis. And then the x-axis will be the dye intensities. And then I'll differentiate between untreated and treated by the color.
Okay.
So dye intensities are 15, 30, 45, 60, and 75. And then the percentage of cells is going to go from 0 to 100 by tens.
Okay.
Yeah. So for treated cells, there's corresponding with dye intensity 50, there's 5% of cells in this. So, then corresponding with dye intensity 30, there's 60%. Sorry, there's 2%. Dye intensity 45, there's 0. Dye intensity 60, there's 93%. And dye intensity 75, there's 0%.
Okay.
And yeah, so basically I just did a bar graph because it would be all weird if there was a line, because there's pretty much 0 here, and then it shows that there's 93% in that area.
All right.
So for untreated, again, we're going to start off with 5% and 15. 30 has 60%. 45 is going to have nothing again. 60 is going to have 35%. 75 is going to have 0. So, yeah, basically that's the graph showing dye intensities with percentage of the cells that are in those stages. Yeah.
Okay.
And then the second question is, at what stage of the cell cycle does the drug act on tumor cells? Let's see if I have this. Change this camera angle. There you go. At what stage of the cell cycle does the drug act on tumor cells? So the first thing that I did when I looked at this was just determined the DNA content per stage.
Okay.
So D1, it'll just be like, it'll just be like 1 times what it normally is, so that's like sort of the baseline. Then S, the DNA duplicates, so it'll be twice as many as in G1. G2, there's still going to be twice as many. Prophase 1, metaphase 1, anaphase 1, 2. And then after telophase 1, it's going to split up, and it's going to go back to 1 times as many, because the cells have divided and essentially have their DNA content
Okay.
as before. So it's still 1. And then in the second stage of meiosis, they're going to divide again into 4 daughter cells. So it's going to be half as much DNA content as it started with, and a quarter as much as there is after it replicates after S phase. So at what stage of the cell cycle does it act? So the first thing that I looked at, since the treated cells are the ones that are acting, I corresponded the treated cells here, since 93% of them are corresponding to dye intensity 60.
Okay.
I figured that they would be acting in dye intensity 60 because that's when they'd be used and they'd stay in that phase.
And what do you mean by that?
So they would be acting in this area when the DNA is proportional to that number, to 60.
Okay.
So then, since it says the more intense the sample is, the more DNA contains, this is the highest amount of intensity, and therefore I figured it'd be the highest, it corresponds to the highest amount of DNA content in the phases
Okay.
that I have right here. So,
I gotcha.
so and the reason why 45 and 75 is 0 is because I figured that this would be the base amount, which would be in G1, which would be 1 times. You never get 1.5 times that in any stage of the cell cycle, and then you never get, what is it, 2.25 times that either. So all you're going to get is 1 times, 2 times, and 0.5 times. 0.5 is during cytokinesis, after cytokinesis too. And so when it says at which stage of the cell cycle it does it act, since 93% are treated acting in 60, and 60 is the highest DNA content when it's 2. I thought it would be acting starting after S phase ends, so end of S, because that's when there's 2 times the amount of DNA concentration.
Okay.
And then it would go from there to telophase 1, which is when right before it splits again.
Okay.
Does that make sense?
Does to me. And then why then do you have a couple here?
You have a couple here because they're still in the cell cycle. I don't know. They're just going until they're gonna reach this phase, and then they're
Okay.
gonna act here.
And then with yeah, that makes sense. So yeah, no, I think you explained it well. Actually.
Okay. All right.
Graph looks good.
Yeah.